Hello, my name is Josh Ruppel. I am a professor of chemistry here at USC Upstate and the ERUPT program director. I wanted to spend a few minutes talking to you about the summer research project in my lab that I'm entitling Development of Thiocyanin Glycoconjugates for PDT. Before we get going, I wanted to spend a minute talking to you about kind of my research group in general. I would say that it's kind of organic synthesis with a slant toward medicinal chemistry. To give you a sense of kind of the types of students that have worked with me over the years, I've had about 30 students with me over the past 10 years or so. This has been a mix of chemistry and biology students. You see some of their outcomes here. 8 of these students have been admitted or completed professional schools. 13 have gone on to graduate studies in the sciences. Several are currently working in science-related fields. I have several publications with student authors. I'm showing here the most recent 2. And also, pre-COVID, we always did some traveling and went to national meetings, and we're hoping to be able to get back into that as COVID willing. But what is my research project this summer all about? Well, again, it's the development of thiocyanin glycoconjugates for PDT. So that raises the question, what is PDT? Well, PDT is photodynamic therapy, which is a treatment that combines a drug, organic molecule, kind of photosensitizer, with light energy to kill a cell. Right? It's a very kind of simplified sort of definition of it. But really what kind of happens here, and I show this a little bit in this graphic here, is that the photosensitizer, we'll call it PS, is excited, and when it's excited, it can make kind of what we call reactive oxygen species, ROSs. And this reactive oxygen species, when produced, can create oxidative stress to the cell and could potentially lead to cell death. And I show here kind of an example of it working on a bacteria, and that's something that we're specifically interested in. Some common uses for PDT include kind of treatments for acne, macular degeneration, like this compound here called Visudyne by Bausch & Lomb. There's uses for this in cancer treatment, like skin cancers, bladder cancers, certain esophageal cancers, and also as an antimicrobial. That's something that we're kind of interested in here. And my group is looking at its potential as an antibiotic. Okay. So why are we focused on thiocyanins? Well, thiocyanins can be used as a photosensitizer, right? And a lot of photosensitizers are really large organic aromatic molecules. Porphyrins are a really good example of this, and chlorines as well. Thiocyanins are related to these. These are a little bit larger, but they still are very large aromatic compounds. Why we're really focused on thiocyanins, because if you look at this UV-vis spectrum, you notice that the thiocyanin here absorbs really strongly in this area that we call the wavelengths of clinical interest. Because that's where the wavelengths of clinical interest is where we get the kind of the maximum penetration depth of the light energy into tissue. So this allows for a really good potential for the thiocyanins as a photosensitizer. There's some drawbacks though with thiocyanins. They can be not very soluble. They can be sometimes very challenging to synthesize and derivatize. But that's something that we're interested in exploring. We're specifically wanting to make thiocyanins that are glycoconjugated, and that is attaching a carbohydrate like the example I've shown here, with the idea that the attaching of carbohydrates will help us improve solubility or bioavailability of the compound. And depending upon the nature of the carbohydrate, we may get some secondary effects here where it could potentially increase the selectivity toward a target. And that's something that we're interested in exploring kind of in future projects. Right now, one of the projects that we're really focused on is trying to develop methods to synthesize thiocyanin glycoconjugates. Right? Now, this is some work that was done by by students last summer as part of the ERUPT program. This is T. Gray Presley and Coleman Walker's work, and they synthesized one of our first examples of a glycoconjugated thiocyanin. You can see kind of their little shorthand here, they call carb or carbohydrate. But you notice it's really low yield. Say that it's crude because really wasn't very pure. But there's really good evidence that we form this compound by NMR. You see here the little picture here of showing the compound is really highly colored compound. It's got this really interesting kind of bluish, greenish sort of color to it. So we're wanting to try to build upon the successes that Tigray and Coleman had last summer and to continue to develop this method. And that can be in the form of trying to increase this process, the effectiveness of this process to make the thiocyanate through their methods, or to continue to explore potentially other methods where we could make the thiocyanate first, then add the carbohydrate to make the thiocyanate. That may or may not lead to higher yield, easier purification. We don't know. These are some of the questions that we want to ask and hopefully answer over the summer. And also there can be a very different sort of method to be able to make the thiocyanate initially. And again, not only do we have these methods that I show on this slide, but there might be other ones that we want to explore as well. So this summer I'm looking at bringing 2 or 3 students into my research group to work on these projects. If you're interested in this, please feel free to reach out to me. My email address is at the bottom of the slide, and I look forward to speaking with you soon.